Antigen specificity. Antigen specificity is the ability of the adaptive immune system to recognize one particular antigen while ignoring others. This mechanism can be compared to a lock and key. In this analogy, the two components have unique complementary shapes. An immune response is triggered only when these two parts fit perfectly.
One example of a molecule with antigen specificity is the B-cell receptor, or BCR. A BCR is an immunoglobulin on the surface of B cells. Each B cell has a unique BCR that recognizes a specific antigen. For example, a BCR for the influenza virus usually does not bind to other viruses. When a BCR binds to its antigen, the B cell is activated. With help of T helper cells, it becomes a plasma cell. Plasma cells produce antibodies with the same antigen specificity. T cell receptors, or TCRs, also have antigen specificity. They recognize antigen peptides on MHC molecules of infected or unusual cells. For example, a TCR specific to one of the influenza peptides responds only to cells presenting that peptide.
B cells and T cells generate diversity during their development through gene rearrangement, which joins selected segments of their BCR and TCR genes.
This process creates diverse receptors with specific antigen recognition, enabling them to recognize an enormous variety of antigens.
T cell specificity is refined in the thymus through two selection steps, positive and negative selection. In positive selection, T cells are tested for their ability to recognize self-MHC. T cells binding self-MHC weakly are allowed to survive. In negative selection, T cells that react strongly to self-antigens are eliminated. These steps ensure that mature T cells are both functional and self-tolerant.
Once a lymphocyte encounters its specific antigen, only those matching cells are activated and expand into many identical clones. This amplification enables a strong targeted immune response.
Antigen specificity plays a key role in helping immune cells recognize and target pathogens accurately, providing precise defense against infections. It also helps immune cells distinguish between self and non-self, so they attack only foreign substances. And it enables immune memory, allowing the body to respond faster and stronger when the same antigen invades again. Without antigen specificity, the immune system fails to recognize pathogens, allowing infections to spread before it reacts. And it couldn't distinguish self from non-self and might attack our own cells, causing autoimmune diseases. Without antigen specificity, the immune system forgets past pathogens and reacts slower and weaker each time they return.